Hello, everyone. I want to present my presentation on computational tissue dynamics of vertebrate embryogenesis. To understand embryogenesis of vertebrates, we use different animal models. Here we have the zebrafish, is the most well-known model for vertebrates, one of the best known of them. And you can see the development of the embryo from the zygote to the late stage development, and through that you go through different stages like the cementation stage, then you go through the dome, the pivot, and then the gastrulation. And the gastrulation is the process when you really give shape to the dorsal side and the vertebrate shape of the embryo. And from then it's more the cementation at the summit stage. The cementation, what I mean by organogenesis, is after 12 hours and 14 hours. Between 6 hours and 12 hours, you have the gastrulation. Is what you can see in this part here from, it's characterized from the epibole here. You see that? Okay. You can see that in a digital embryo. Digital embryo is when you image an embryo of this is the gastrulation process, and then you see the embryo developing. This is a zebrafish embryo that has been imaged with microscopy images with 2-photon confocal microscope. And then you have the staining for the nuclei, which now is looks here in green, and then the membranes that look in red. And with this staining, you can make fluorescence images and make the images along time in 3D, and then you have a four-dimensional image or a 3D plus time image if you want, which is different volumes along time, and you can create this sort of movie. Along this, you can make this digital embryo, and the best thing that you can do with this is to reconstruct the cell lineage. Is the movement of one of these cell nuclei that you can see here on the right, and then you can cement the cells, and then you can create the lineage, which is the position of the cell along time and its divisions. And then you can create this digital embryo like an atlas or like a digital representation of the embryogenesis instead of an image, more digital. And with that, we are able to understand more the mechanics of the embryo with the digital lineage. And here we have different types of movement of cells, like our very well-known, well-studied movement of cells is the contraction and the stabilization. It's contraction and expansion. And then you have actomyosin elements playing here because you have the actomyosin activated, and then it produces the contraction. And when the actomyosin changes the process, it goes to stabilization. It's an expansion, and then it's contracting and expansion, so that the cytoskeleton of the cell is changing, and then it's propagating the mechanics of the cells to the tissue. It's a very important concept. It's a multi-scale process, and you can see here the patterns, because some cells are contracting, others are expanding, and what is happening is that you see patterns, and these patterns have a tissular level. So what we want here is to use the cell lineage. It's the trajectory of the cells along time to understand the mechanics of the embryo. And as I was saying, this is a multi-scale system. It goes from the component level of the cytoskeleton to the whole embryo, and it goes to the scale of analysis that we are interested in is the small domain, because it's the relation of the cell with its neighbors, the cell and the cell neighbors that you see here, and the small domain, and it goes to patterns to the tissue domain. And this is the cell dynamics. What we understand as cell dynamics goes from the cell, but it grows up to the embryo level, because through the small domain and the tissue, you can create patterns that give shape to the embryo. And then it's how you go from the cell lineage to a multi-scale representation with different length scales, because we will be able to make an analysis of the different temporal scales, not only spatial, because spatial will go from the cell to the embryo, but also in time, which are the scales in time. And see here we have several movies of different embryos. We have here the embryo was seen before, and we say here how another different embryo looks like, which is contracting the cells. So here we have a cell tracking in the left, but here we have another type of representation, which is the deformation of the cells. This is two different scales, the tissue level at the right and the embryo level at the left. And it's a different type of description of the mechanics, because in one is sparse because you have several cells in the tracking, and in the other way you need a representation of the cell contraction. And what we propose here is an Eulerian and Lagrangian framework to analyze the kinematics. And how you make this, because you analyze the displacements, you noise the filtering, you make a spatial filtering, and then you make a flow representation of the dynamics. And then you can get different type of tensors and descriptors. This is the workflow that we propose for the cell lineage. But if you're analyzing at the tissue level, the information is different. You have to image the filter, and you make another strategy that is B-splines registration. And with this, you have a dense description of the tissue. And then you can have another type of tensors also to make a Lagrangian analysis. A Lagrangian analysis is a Lagrangian from the perspective of the tissue. It's not an spatial representation from an external observer, it's internal observer. And from this way, you are able to see the cumulative deformation of the tissue along time, and you can start different types of temporal descriptions. And this is very powerful to understand the mechanics of how we can go from different stages of development. So this is the different, we go first around here, the different descriptors that you can obtain when you are analyzing the velocity field of the cells. You analyze these descriptors, compression, rotation, and distortion, and also the topology of the deformation. You can see here the different topologies. And if you want to go into a cumulative deformation along time, you have another type of description that is also compression, rotation, and distortion, but there are different descriptors. And you can see it here that you have how a small domain evolves that can be expansion, and you see the expansion along time. It's not between two, it's not spatial, but it's through time because it's Lagrangian. And then this is the kind of analysis that you can do Lagrangian, because you have the trajectory and you see in each time point the cumulative deformation respected to the initial point. And you can do different type of rescaling of the descriptors. So what you have at the end is that you want to measure the different scales and pattern along the different stages of development. And we can do this as a Lagrangian representation seen from the cell perspective. This is when we understand for Lagrangian. And for instance, we go into the results. Here we have 5 different embryos doing the gastrulation. You can see that it's kind of similar process, but it's not always the same. You need to get into more details, quantitative details on how these different embryos are different. And then we have here the descriptors. You see the rotation, the expansion, the velocity, and we can quantify with colors and with numbers, which are the different level of expansion and compression of the tissue a long time. It's not just a picture, but a whole movie of descriptors. And then you can see how it evolves also across the dorsal side. You see how the dorsal side is created with a tissue that is the invagination of the gastrulation, and you see all the different patterns in this time. Is corresponding to the cell stage and through all the pivoli. You see the pivoli is the tissue that is underlying the endoderm. The mesoderm is going behind, and then you can see this in through time in different types time snapshots. So between 6 hours and 12 hours, the gastrulation is achieved, and we can see here as a columns the time, as a rows the time, and columns the descriptors. And we see how the compression, for instance, the descriptor P in the middle is changing, and you have the different embryo level, and from the tissue analysis, we see embryo level patterns that is giving shape to the embryogenesis. So the conclusion here is to analyze the embryogenesis is not only a chemical process of the actomyosins, it's a communication of different cells in the cell domain level, but the cell domain level scales up and makes a pattern that is from tissues, because you have different tissues interacting, but you have also embryo level. And this embryo level patterns is what is giving shape to the vertebrates. And you can see also in time to the Lagrangian descriptors how the patterns flow with the embryo. So you have expansion here in the J descriptors, the second column. And then you can see in red, this means that it's compression, but the compression is going up. So the compression is propagating. It implies that it's a shaping process. It's not only spatial, but the cells along the time are experiencing different type of stimulation. And this is different to the concept of another type of process, like in the dorsal cluster, which is not a vertebrate, that is a contraction. And it's a tissue level, but it's not embryo level. Because if you see here the relationship between the cell deformation and the actomyosin, that here the image below is actomyosin, is myosin market, and you see a different Fauci concentration of myosin. It's giving rise to a pattern that is, you can see here at the tissue level, but it's not at the embryo level. So for the vertebrate generation, you need to have patterns at the embryo level. So the conclusion is that you can approximate the cell lineage as a flow, and then you can get Lagrangian maps along time to see how the cells have an experience of deformation. And with this, you can observe that there's some kind of fluid behavior of the embryo to give shape to a whole embryo pattern instead of a tissue pattern. And with this kind of embryo level pattern, you are able to give rise to the vertebrates. If you don't have this embryo patterns level, you, as we were seeing here, as we were seeing in this figure, and in this figure, you cannot create the dorsal side, the vertebration, the vertebral structure of the embryogenesis. So this is the main conclusion that you can extract when you analyze patterns from the cell lineage. Any question? Thanks.